I mean, when we are thinking of plasticity, I always also admire this idea of ploidy, for example. So you
Mm.
mentioned that planaria are mixoploids. Then there are, I mean, we can talk about plants itself. We know that they change their ploidy. So how much that has to do, or do you think that this can be also a factor?
I don't know much about it, but I will tell you my favorite ploidy story that I think bears on this.
Hmm.
So salamanders, uh, you can there's a little tube that goes to the kidneys, the kidney tubule. If you take a cross section through this kidney tubule, what you see is typically 8 or 10 cells working together to make this nice kidney tubule. Okay. Um, if now one thing you can do is you can make these newts that are multiple ploidy, so you can make 4n, 6n, 8n newts, right? So the first amazing thing is that you can double or triple the amount of genetic material. They don't care. Fine, you still get the same newt. So that's kind of that, all that speaks to my same point, is that it doesn't really assume. So embryogenesis does not assume how many copies of your genetic material you have. So that's the first thing. The second thing is, as you increase the ploidy, the cells get physically bigger and bigger. Okay.
Yes.
When the cells get bigger and bigger, the salamander stays exactly the same. How can it be? Because fewer and fewer of the larger cells are being used to make the various organs. So you can't count on how many copies of the genes you have. You can't count on what your cell size is. If you're even a human embryo, we can cut you into 4 pieces and you get full quadruplets. You can't even count on how many cells you have. Now the ploidy story is so you really just assume. You have to, as a proper embryo, you have to assume you don't know much going in. Who knows what things are going to be like, and you still have to be able to do the right thing. Now here's the rest of the ploidy story. So when you make 8n newts, the cells get so enormous that there's not room for more than one cell to make a tubule. What do? A single cell bends around itself like this, leaving a hole in the middle, which gives you the tubule. Now so the thing that's crazy about that is that now that's a completely different molecular mechanism. Instead of cell-to-cell communication, which you need to make the tubule, you are now using cytoskeletal bending to drag yourself into this crazy shape. So what I love, right? It's amazing. And so what I love about this is that it's a good example of top-down causation, meaning that what you've got is in the service of this anatomical feature, different molecular mechanisms get called up. And we're seeing something like this in some of our not yet published stuff in the Xenobots too. Different molecular mechanisms get called up to do their thing to achieve this sort of higher level outcome. So, so I think ploidy is a fantastic example of this, because if you don't assume how many copies of your instructions you're going to have, you don't assume how big your cells are, you don't assume how many of them you have, you better have rock solid morphogenetic control to get the job done despite all of that. And salamanders do, which is why they're so regenerative. But they're not as good as planaria, but they're pretty darn good. They regenerate all kinds of organs. So
I mean, underpinning all of that, this cell volume and ploidy, it's not just ploidy, it's genome size, it's the total amount of DNA in the nucleus. So it's the nuclear volume. There is a very tight correlation between nuclear volume and cell volume, which used to be known as the C-value paradox, which is to say the cells, mostly amoeba, have some enormous genomes, thousands of times larger than the human genome. Why? It's, this whole idea of junk DNA. What are they putting in there? A large amount of it is that you have a large cell, you have a large nucleus, and you fill that nucleus with DNA. And ploidy forces you to have higher ploidy forces you to have a larger nucleus, which forces you to have a larger cell, and that forces you to be resourceful in terms of, if you want to make a tubule, you do it from a single cell instead of 14 cells or what it used to be. So I don't think there's ever been a good explanation for why a large cell would have a large nucleus. There are ideas out there, but they often relate to the ratio between the number of ribosomes and the number of nuclear pore complexes and this kind of thing. None of them are particularly persuasive. I don't think we have a good explanation for why a large cell should have a large nucleus, and yet it's able to force all of these changes on you.
So when you mention about neurotransmitters, which is like one level down kind of regulation, let's talk about anesthesia, because I think that's also something which is one level down, regulates it. I don't know how much of the work is done on some sort of simpler scales, but okay. I mean,
Well, you
let's
can
talk about it.
anesthetize single-celled critters, planaria, and things like that.
Yeah.
I mean, I discussed in the last chapter of the book some of the work from Luca Turin, showing that anesthetics affects electron transfer to oxygen. And we've been doing some work on that just recently. We've not published any of it yet. We've not got very far at all, so I won't say much. But we've basically we also find that it lowers oxygen rate of consumption of oxygen, and depending on the context in which you give them, affects the membrane potential. Now, to try and distinguish between just the mitochondria making ATP and that ATP being used to control the a plasma membrane ion pump, for example, an ATPase in the membrane. Obviously, if you don't have as much ATP, that pump is not going to work as well. And so, this is not a causal link between mitochondrial function and consciousness. All it says is that underpinning this energy-consuming process, which is consciousness, energy matters, which is a statement of the obvious, I suppose. But there's also a possibility that there are direct fields involved, that you're influencing the electromagnetic fields generated potentially by mitochondria, and that there can be direct interactions with the plasma membrane field that way. That's a much more exciting possibility to my mind, but very, very difficult to distinguish between how do you change the field in a measurable way while maintaining ATP synthesis at the same level, which is a tricky thing, a tricky mechanistic thing to do. But we're trying to get at that. I mean, I think this is perhaps one of the biggest questions in science now, is what actually is going on in the wiring of consciousness, if you like. And I'm certain fields are involved, but how exactly, I'm not so sure.
Yeah, so it's really interesting. You might enjoy. I've got this book from this guy, J.C. Bose, Bose, and I can send you a PDF of it. He's an Indian guy from over 100 years ago that was doing a bunch of physiology, and he started anesthetizing animal tissues and using electrophysiology to monitor what happens, and then he did the plants, right, and then eventually a metal bar. So he's got this book called Response in the Living and Non-Living, where maybe so I don't know the mechanisms that underlie any of that, but there could be, if it's the kind of thing you're talking about, I could easily see why he would be seeing effects like this in inorganic substrates as well.
Yeah, I tend to draw a line at life, which is to say what I was talking about at the beginning about bacteria using the fields on the membrane as a means of integrating the metabolism of the cell. And I would see then the central nervous system as some kind of an amplification and far more subtle and complex, of course, but still somehow linked to the integration of information through linking your metabolic state at a cellular level and then at a network level and so on. Whether you need fields to be involved in that, I don't know, but we can measure changes in the EEG. And I don't think we have an absolutely clear idea of exactly what is generating the EEG at a subcellular level. Is it just the plasma membrane, the axonal membranes that are depolarizing, or mitochondria playing a role in that as well? So that's an area I'm fascinated by, but it's difficult to get at.
Yeah, so in the I think if we think of the same about the same topic, interesting thing is about the synthetic cells or synthetic life in general, like all the work which is going on there. I mean, from the engineering perspective, right? That's also kind of let's say crucial in a way, or it is focused on the information centric view, right? Like people are trying to put together again some sort of genomes, some sort of genes, transcription, translation. Of course, they have membrane in, but my question, or maybe briefly, if you can just explain your views. So how important would be this kind of bioelectric or kind of this software that we have discussed today for to create those kind of synthetic systems? Maybe
I mean, a one-word answer: all important. I would say. I don't think you're going to do it without.
Like, how do you
100? Yeah, 100.
Like, like how, or is it possible to kind of I don't know, do it without the information part, without the hardware?
I think you can only go so far without genes at all. How far, I don't know. I mean, the planaria is a very good example there. You can scramble your genome, but presumably you can't completely collapse your genome. When I think about it the other way up, with the origin of life, where does information come from in biology? And I'm fairly persuaded now from our own experiments, but other people's work around the world as well in the last 5, 6, 7 years. A lot of metabolism is spontaneously self-organizing. You start with CO2 and hydrogen, and you get Krebs cycle intermediates. You get amino acids, the same ones that we have in metabolism. You get nucleotides that way as well. We've not done them all yet, but we've done some, and other people have done others. And this idea then of a spontaneously self-organizing metabolism that starts with CO2 and hydrogen and gives you all the monomers and eventually polymers. It's by no means is any of this proved, but 5 or 6 years ago it would have been complete hand waving, and now there's some experimental backing for quite a lot of it. And you think, okay, well, it's going to make trace amounts of things like nucleotides. How can it reliably make more nucleotides and get better at making copies of itself in the absence of genes or any information? And the answer is, well, positive feedback loops will allow you to kind of feed off the environment and off yourself to get better at making copies of yourself. But you're only ever going to you're going to keep repeating the same network under the same conditions. If you change the conditions, you might get a different network. It may or may not function. But under this set of conditions, you will keep recovering that same network until you introduce genes and information into it. And you can into that world, you can introduce random sequences of RNA, and they will have directly or indirectly some effect on the rates of cell growth and flux of metabolism. Even just introducing a piece of RNA is actually going to change the metabolic rate, because you're now polymerizing nucleotides. So you're pulling nucleotides out of solution and you pull metabolism through faster. So even making a completely random piece of RNA makes you better at doing what you were doing before than if you hadn't done that. So there's kind of interesting insights into what is information in biology, where is it coming from? And it's what it seems to be allowing is effectively plasticity, which is to say you've got this hardwired proto-metabolism, which is basically thermodynamics and kinetics, and it's, you will go on recovering exactly the same thing. What genes allow you to do is just change it a bit and allow a different outcome, and that outcome can be reliably inherited. And by the time we're getting into networks of cells, then it's interesting, because what I'm taking from what Mike has been saying is that the genes are becoming almost buried again, and it's the network of the cells which are generating higher level properties, which are forcing them to behave in a similar way in a similar setting, and a potentially completely different way in a different setting. So there's this interplay all along between kind of the hard wiring and the plasticity and the information, which means different things in different contexts, but allows the plasticity, really.